So, good morning, everyone. Welcome to the first edition of the WhatsApp with Malditel mass spectrometry and microbiology. I'm Maureen Faucherolles, and I'm a PhD candidate at the Luxembourg Institute of Science and Technology, and I will be your host for today and tomorrow's session. So we already have a lot of people present today, and we're pretty international, in fact. So before to start, I would like to say a big thanks to all the presenters of today. Also, for your information, the session of today and tomorrow will be recorded. And so at the end of each session, you will just receive an email with a link where you can rewatch the full session. Also, the email will be attached also a survey from the presentation, so we can improve our next events. So our today's agenda is pretty full. So we will have 5 presentations with 2 keynote talks. So today's presentation will have a main focus on antimicrobial resistances, machine learning, and also current application and the future application of mass spectrometry in routine clinical microbiology. So let's jump into it. And so I would like to introduce our first speaker, so Dr. Belen Rodriguez from the Madrid Hospital. So as a quick presentation, Dr. Belen focused on the implementation of the MALDI-TOF mass spectrometry in clinical microbiology laboratories since 2011. So upon identification of wide range of microorganisms, our group has applied machine learning techniques for classification of microbial isolate resistant to antibiotics or antifungals, or belonging to different sequence type. So Belen, the floor is yours.
Yeah, thank you very much, Maureen. Thank you for inviting me. Thank you for giving me the opportunity to talk in this introductory talk today. I will talk about sorry. Oh, sorry. So, I'm using the screen now, right? It's me.
Yes, we see it.
Okay, I cannot just pass one by one. It goes to it goes directly to the second one. Okay, thank you. So I'll just try to summarize what has been happening with MALDI-TOF and its role in the clinical microbiology laboratory. And so it's a really amazing story about how this instrument was a completely unknown machine, and now it's a key machine, a key instrument in our laboratories. So I'll give you a little bit of a background, and then I'll talk about the identification of bacteria, which was the first task that was assigned to this computer, to this machine, and then we'll talk about how the difficulty of the identification was increased, and identification from difficult microorganisms was a say. I'll elaborate later what I mean about difficult microorganisms, and then I'll talk about how the MALDI-TOF was applied to direct identification of pathogens from clinical samples, the detection of different resistant mechanisms, and bacterial typing. I'll just talk briefly about each of the topics, because real experts will elaborate about these topics later. So first of all, I want to talk about one of my favorite characters, my favorite roles in this amazing story about mass spectrometry and its application in clinical microbiology. And it's Professor Catherine Fencelau. This paper from 1975 described the identification of bacteria using mass spectrometry. It's true that at that time, it was pyrolysis mainly what was applied, and this technique completely calcinated protein. So it was small peptides, as far as I understood what was analyzed. So it was 10 years later with this other amazing character, this developed soft ionization, which allowed the idea of Professor Fencelau to be really carried out, to be applied in the laboratory. If I strongly suggest, or I encourage people to read it, his lecture during the Nobel Prize ceremony in 2002 when he was awarded this prize. So both characters, both researchers established the basics for the identification of bacteria or other pathogens or their microorganisms using MALDI-TOF. So this picture I like so much. It's both of them during the 2002 Nobel Prize ceremony. So once this technique was established and it was assayed and it was shown to work, there was a boom of MALDI-TOF from 1995 to the present day. This a search in PubMed made a couple of days ago with these keywords, and the results is more than 3,000 articles about identification of different microorganisms from bacteria to yeast, fungal isolates, filamentous fungi, mycobacteria, whatever pathogens you may be thinking about. So there's, as I said, more than 3,000 articles about how Molotov identified, and so there was a huge change from the conventional biochemical techniques that were applied before and mass spectrometry. So shortly after the development of this technique, there was a huge number of really impacting papers. This is a Nature Biotechnology paper, 1996, about how this procedure was applied in the laboratory. Shortly after that, there was this other paper from Professor Fencelau describing the identification of bacteria with mass spectrometry and its comparison with different databases. Amazingly, in 2000, there were already papers describing how Staph aureus, methicillin-resistant Staph aureus could be detected using MALDI-TOF from intact cells. And this other paper about phyloproteomics is from 2001. So there was a huge development of this technique and a huge improvement and application in the laboratory. And for me, it was also this paper which kind of it is for us the starting paper, or the basics about how to implement MALDI-TOF in the laboratory, and how it could be applied to different identification of different bacteria using a very easy methodology. So you all know how to perform MALDI-TOF, but just to tell about little details about how the techniques work. This is how the manufacturer planned that this should work. That from couple of colonies from a plate, you could spread it on the MALDI plate and add just 1 microliter of organic matrix, and that would be enough for the laser to ionize and analyze. It was shortly after this basic methodology was released that some researchers just found out that formic acid could perform some on-plate protein extraction and improve the identification. So this is mainly what most people use in their labs. Just spread the colonies, add 1 microgram of formic acid, let it dry, and then add the matrix, again let it dry, and the sample is ready to be analyzed. So the whole procedure takes less than 5 minutes. And the longer this methodology was applied, the better the identification were achieved, and the bigger the number of different species were identified correctly. So from gram negatives that were probably the first and the easier to be identified with Molotov, to gram positives and and anaerobic bacteria. I'll talk about them a little bit later, and the mycobacteria. So difficult to identify isolates. So that's why I want to talk about like 3 different groups of what I call difficult microorganisms, or those that are not that easy to identify in the laboratory. And I think each of them taught us a different lesson. So the first one is the group of the anaerobes. So in this case, what we learn is how important it is to collaborate with the manufacturers. There's this ENRIA project that was launched from the European Study Group for Anaerobes, the SGI group. So they collaborated, different laboratories from all over Europe collaborated with Bruker, and they collected like 6,000 anaerobes from different laboratories, and with those they managed to implement, to improve the number of these isolates in the available databases. So it meant that after this amazing amount of anaerobes were included in the database, API tests were not used anymore for anaerobes. They were not needed, and Malditel was included as one of the reference techniques for anaerobe identification in this paper about how to identify anaerobes. So now it's like officially technique or a recognized technique for anaerobe identification. So for me, the second group is the mycobacteria. In this case, the lesson was united we started, united we can improve things. So this is just an example of how mycobacteria were identified when we started working with Malditel. But and in this case, it was the companies, different companies that improved this group of microorganisms so well that in no time most of the species were covered in the databases. So this the table at the bottom, this shows just a few missing species in the among the more than 200 mycobacteria species that are described so far. So different groups just collaborated and just try to standardize the known methodology for the identification, but it was more from the laboratory side, since the manufacturer had, different manufacturers had done their homework. In the case of fungi, it was just the opposite, that no databases were available that included clinically interesting species. So in some databases that were available, references from fungal isolates were made from fungi growing in liquid media, which is used for research, but not so commonly for routine. So this was not very helpful. So in this case, different laboratories just made the effort to create reference protein spectra, and they built their own in-house databases. And it's outstanding. I want you to show here this web application that this group from Dr. Hendricks and Dr. Pierrot just launched a couple of years ago, that is very very, at least in Spain, many laboratories use it because it's very easy to upload your protein spectra and compare them with their database, the online database, and get back the identification. I tried to use it, and I keep the link here, but it didn't work last time I tried. I'm not sure if it's not working anymore or if I did something wrong. So this with this philosophy of sharing and uploading things online so many other users can get access to it. Recently, we found out about this MALDI-TOF database, and I wanted to share it with you. And then I found out that Björk will be talking about it later. So just let you know that this tool is available and it may be useful in certain circumstances. Yeah, regarding direct identification from clinical samples, for me, this is one of the most impacting application of Molotov. So instead of culturing the broth from positive blood cultures and wait 24 hours or 18 hours until next day, different techniques, identification techniques were performed, we can just directly from the broth apply different methods, certification, ammonium chloride. There are three, as far as I know, there are 3 different commercial methods that are also available, and identification is obtained in more than 80% of the cases in less than 30 minutes, which is really, really impactful. Also from urine samples, there's different papers published. In this case, if you culture the urine overnight and you get the colony, next day you can perform MALDI-TOF and get more than 90% correct identification. That is what we do, and I think this is practical. But anyway, different articles, different authors have published how MALDI-TOF perform from urine samples, and in most of the cases, as I show here, this paper from Burillo et al., 82% of samples were identified. For me, the main problem from this protocol is that it's long, it takes time, and usually patients are not so severely ill patients. I mean, those with urine infection. So you can, in our practice, in our experience, we keep this protocol for very urgent samples. Otherwise, we skip this very, very long procedure and wait until the colonies have grown on the agar fluid. But it works. That's one nice thing to know. And yes, just wanted to show this paper from Bishop and collaborators. They tried 44 CSF samples in cerebral spinal fluids. So gram negatives could be identified from these samples in 17 out of 21 cases. It is not so easy from gram-positive cocci, only 1 out of 11, and they couldn't identify yeast in these samples. But it's kind of promising that gram-negative rods were identified. No later paper that I know of has been published, but yeah, it's good to know that you can try directly from CSFs. Just very quick. Couple of things about the detection of resistance mechanisms, because Dr. Oviano will be talking about it in depth just right after me. So the 2 approaches for this method is different protein spectra from organisms with and without resistance mechanisms can be compared, and so for a quick detection or discrimination of those populations. And hydrolysis is also applied in the case of microorganism hosting beta-lactamases and carbapenemases. In the first case, spectra look like this, like one population, in this case, Enterococcus faecium with the Van B mechanism, has some display some peaks that are not present in the sensitive isolates. In the case of hydrolysis, some peaks are specific of the intact antibiotic, and they disappear or change a little bit when the bacteria hydrolyze them. And regarding bacterial typing, that's a very interesting application of MALDI-TOF. This paper from Soget and collaborators, they established the workflow that should be applied for this purpose. And for us, it was also very important to learn from this other paper from Overlay and collaborators, where they show how technical and biological reproducibility should be assayed in order to apply MALDI-TOF for bacterial typing. So in this case, what we learn is that we need to assay the technical reproducibility, which means just to compare duplicates from the same sample. So in this case, we apply the same microorganism onto two spots, and we get at least four spectra from two spectra from each spot. And usually we have second set of two spots and do the same. So in the end, you have different spectra from the same isolate, so you can compare and get an average spectra of one representing that sample that you're testing. The second thing is the biological reproducibility. So in this case, what we want to know is if different subcultures of the same sample provide the same kind of spectrum. So usually we have one colonies from day one and do, as we showed above, different duplicates. Then we subculture sample, and on the day two we do the same in day three, and then we compare different protein spectra from different days. And then ideally, then you test the same samples in different research centers in different laboratories and compare data too. So just want to show you a couple of examples that we have performed in our laboratory. This is not strictly bacterial typing, but we try to differentiate species from within the Cryptococcus neoformans complex, and also hybrids from two different species, and we could reasonably well separate those three populations using MALDI-TOF and different machine learning algorithms with the same samples using a supervised SVM algorithm. This is a recent paper still under review in order to differentiate Aspergillus fumigatus species. These are sensitive Aspergillus, and these are resistant isolates, resistant to different azoles. This figure here, we show that we can differentiate resistant from susceptible and from cryptic Aspergillus from different cryptic species. This is a very recent study we haven't finished yet about discriminating species from the Enterobacter cloacae complex. So we have seen that the main or the more common species can be differentiated. And just to wrap up, I just want to summarize that it's important to have one instrument in which you can perform these many different applications. So one MALDI-TOF for identification, AST, and ideally also for bacterial typing. And so if, you can more or less standardizedly apply these three applications. Probably there will be more in the future. So direct identification for clinical samples is one of the main applications that you can also apply using the same machine. That besides it's inexpensive, it's user friendly, and requires very short training and very easy training. So ad hoc software for different applications is coming up. It's being developed, and it will help in the future to perform these different applications in a more user friendly way. and then most probably biomarker peaks for different important groups of bacteria will be developed too, and that will make things easier in the laboratory in the future. So that's all from my part. Thank you very much for your attention. So here's my my email and my Twitter in case you want to contact me. So thank you very much.
Okay, thank you very much, Belen, for this really nice introduction for our symposium. So I didn't say it in the beginning of the presentation, but if you have questions for the speaker, please do not hesitate to write it down in the chat. We will just read it after each presentation for a questions time. So we still have a few minutes for some questions. I don't see any for now in the chat, so I would just ask mine. So you were talking about using MALDI-TOF for typing. Do you think that one day maybe that MALDI-TOF mass spectrometry could replace such techniques like MLST or any genomics, or it could be only like a first screening tools for routine laboratories?
Yeah, for me, first screening would be more accurate. I don't think it would replace MLST or whole genome sequence. Probably it was more like both things together, like complementing ones. And since MALDI it's really inexpensive and it's very quick, could be like more like a screening technique, and only those samples not fitting in the pattern, then you do MLST or other genomic techniques, but not taking over the whole genome sequence or the genomic methods, but complementing. That would be my opinion.
Okay, great. So if there's no question, I just suggest we just jump to the next presentation. And if we have any more question, we will send it to you, Belen, of course, no problem.
Okay.
So let's jump to the next presentation with the Dr. Marina Opinio. Dr. Marina is served as a researcher at the National Health System in Spain, but currently she's working as a clinical microbiologist at the microbiology department of the University Hospital A Coruña. She's been working on research in the field of bacterial resistance to antibiotics, and she has a main focus on the use of the MALDI-TOF mass spectrometry as a rapid technology for diagnostic use. So, Marina, the floor is yours.
Okay, thank you so much, Maureen, for inviting me to be here today. I think it will be a very interesting symposium, and to the audience for listening to me. So, as Maureen said, I will be talking about how can MALDI-TOF help in the detection of antibiotic-resistant bacteria in clinical practice. I would like to start by speaking about how the clinical impact of antibiotic resistance. We all know, and it's already recognized by different health organizations like CDC, that there are increasing rates of antimicrobial resistant bacteria in recent years, and each year there are more and more deaths attributable to antimicrobial resistance. The CDC established the biggest threats related to this resistance, and establish some items like carbapenem-resistant enterobacteria, carbapenem-resistant Acinetobacter, Candida auris, Clostridium difficile, and multi-drug resistant Neisseria gonorrhea as one of the most serious and actual threats. The ECDC also have a map, a Europe map, where you can find the rates of carbapenem-resistant Klebsiella pneumoniae, and you can clearly observe here that in the last 10 years there are more and more countries which are increasing their rates to carbapenem-resistant Klebsiella pneumoniae, especially in the south of Europe, in Italy or Greece. There are more than 50% of this bacteria. There are a lot of tools in clinical laboratories that we can use for diagnosing this resistant. But the most important and the gold standard, of course, are molecular methods. One of the most widely used is the Expert Carba-R from GeneXpert. It is a very easy to use technique. You can just use the direct sample and a swab, or you can use colonies from plate cultures. You have to insert the sample in a buffer, then transfer it to a cartridge and insert the cartridge in the equipment, and after 50 minutes more or less you have a result. There are a lot of published papers evaluating this technology, and all have very good results. Another technique which is very interesting because it's cheaper and it is fast and easy to use as well is immunochromatography techniques. In the most used, I think it's this one from Coris, which you can see in the image, but I think all of them work more or less the same. You can have one target. If, for example, we have only the target of OXA48 because it is the most prevalent enzyme in our area, but you have also multiplex targets in the same cassette. So you can analyze OXA48, KPC, NDM, or VIM in the same bacteria. It only takes 15 minutes to give you a result, and it doesn't need any expertise or whatsoever, so any technician can perform it in routine. We cannot forget phenotypic methods, which are very, very common in all laboratories, like the Hodge test, which maybe is less used now because it has a good sensitivity, but it is not very specific, especially in certain bacteria, as Enterobacter. And the combination disc testing, which is also very useful. Both techniques need the growth of bacteria, so they are slower. They take like 24 hours to give a result. But also on this line, the biochemical test, or also known as the Carba-MP test, is a phenotypic method and gives results very rapidly and is very accurate as well. It is based on a change of color in the medium where you have to incubate bacteria with the antibiotic, and after the activity of the enzyme, you measure if there is a change in the color of the buffer, the reaction is positive for the hydrolysis. In this line, we also have Maldi-TOF, of course, which is the main topic of this talk. It is a proteomic method. It was introduced in the last UCAS guidelines for detection of resistant mechanism and specific resistance, clinical and epidemiological importance. You have here the link. the use that was authorized by UCAST was detection of carbapenem hydrolysis with MALDI-TOF. So the principle is to detect in a MALDI-TOF device the change in the mass peaks of the antibiotic when the bacteria is incubated previously with a solution of this antibiotic. The procedure is very easy. As I said before, you just have to incubate the bacteria with the antibiotic a certain time at 37 grades. You can start with a colonies in plate culture, so you can start directly from the clinical samples. If that is the case, you have to perform a bacterial extraction. But then the procedure just follows the same. You have to incubate and then you have to centrifuge, and the supernatant is spotted in the target place. It gets dry, and you apply it after that the matrix and perform the lecture in the equipment. As a mass spectrometry technique, you measure a mass change. The mass change in the antibiotic. When the hydrolysis reaction takes place, the antibiotic is modified, and the ring is opened, but not only the structure is modified, but it is also produced a mass change. That is exactly what we can detect with MALDI-TOF. So we have a certain pattern for the intact molecule, for the antibiotic in its natural state, and we have a different spectra for different spectra for the antibiotics after they are hydrolyzed. So the first one that started with this approach was Jaroslav Habrak. He published this paper in 2011. And he already used meropenem for his assay and introduced the different mass peaks that were present in the intact molecule in the meropenem and then in the hydrolyzed meropenem, so that everyone could recognize those different patterns and then get to know if your bacteria produces enzymes, so it is able to hydrolyze the antibiotic. More or less at the same time, Catherine Sparvier from Bruker analyzed a series of antibiotics, Beta-Lactam antibiotics, and also just went deeper into this approach. Jaroslav Habrak again made some serious improvement to this assay because we had serious problems at that time in detecting an OXA48 enzyme, because as you already know, they have a low catalytic activity, lower than the rest of enzymes, so we could easily detect VIM or NDM or KPC, but OXA48 were more difficult to detect. So adding ammonium bicarbonate really helped to detect these enzymes and increase the sensitivity from 75% to almost 100%. Our group also was or is still very interested in this type of approach, and we analyzed different antibiotics to see and to evaluate which one was the most interesting for detection of beta-lactam resistance. And we realized that the ceftriaxone, which wasn't used at that moment, was very, very accurate. It gave very good results, 100% of sensitivity, and it was hydrolyzed very rapidly in just 30 minutes. So after 30 minutes of incubation, you can measure and realize and detect if the bacteria carries a beta-lactamase or not. We also try to detect other mechanisms of enzymatic resistance by measuring the structures of the mass peaks of different antibiotics. In this case, we try to detect the plasmid-mediated quinolone resistant determinant. For that, we use 3 antibiotics. We used ciprofloxacin, norfloxacin, and levofloxacin. You have the structures in A, B, and C. So A is ciprofloxacin, B norflox, and C levo. So this enzyme performs an acetylation of the original antibiotic in this position. So we used levofloxacin, which is already methylated in that position, as a negative control, and we really could observe this modification in the structure of the antibiotic. In the figure that's above, you can see in A, this is the native structure of ciprofloxacin. B is the structure of the antibiotic once it is modified by the enzyme. What was curious here was that as it is also a low activity, has a low activity enzyme, we could observe in the same spectrum the mass peaks of the original antibiotic and the modified antibiotic, which was really in line with this low activity, as if you only have this determinant of resistance in bacteria, usually the minimum inhibitory concentration is below the cutoff of sensitivity. So it was a very interesting approach. We also try to apply all these approaches in indirect samples, as I think it is the most interesting application after all. We try to detect Beta-Lactam assays directly in blood cultures, and we had very good results. We also performed the assay directly from urines, which also gave very good results. We compared the usual workflow that takes place in our laboratory, just culturing and then at the following day performing the antimicrobial susceptibility testing. And we compared to perform a bacterial extraction from urine. We perform it with a sepsitide kit, a modification in the conventional procedure for blood cultures, and then perform the carbapenemase hydrolysis assay using imipenem. And we had 91% of accurate identifications and 100% of sensitivity in the detection of carbapenemases. We just had to introduce a first step of screening of flow cytometry, because it is true that if the concentration of bacteria is low, it is very difficult to perform the hydrolysis assay. There is an automated software from Bruker that performs the interpretation of the spectra automatically and gives you a report where you can check if bacteria hydrolyzes or not the antibiotic. It is commercialized with 2 kits, the Carba kit and the Cephas kit, which you can already buy and use in your laboratories. It is helpful because it doesn't need any expertise in mass spectrometry. You just have to follow the recommendations that appear in the kit and perform the assay in the equipment as you perform any identification. What are the main advantages of MALDI-TOF? In my opinion, one that it is very interesting is that in recent years there are appearing more and more variants of carbapenemase enzymes that lack carbapenemase activity, as for example KPC28, OXA48 variants like OXA163 that lack this activity, carbapenemase activity. these enzymes usually provide positive results in molecular assays like conventional PCRs, like GeneXpert, or even in immunochromatography assays. And the advantage of MALDI-TOF is that it measures the real activity of the enzymes. It provides rapid phenotypic results with high sensitivity and specificity, and it's really very cheap. It is true that it has also disadvantages like the hands-on time, which usually it takes more time to perform a MALDI assay, and it requires certain knowledge in spectrometry, especially at the beginning when you set the assay, you standardize the assay. In our case, what we perform in our laboratory, we kind of use all techniques really, because I think no one can replace another, so they are all complementary techniques. But mainly, when we have a suspicious of a bacteria carrying a carbapenemase, we perform in the first place an immunochromatography assay. If it is positive, we just confirm the diagnosis, because we don't have any OXA48 variants in our area, and we have a very high prevalence of OXA48 enzymes, so we just use one target. If the result is negative, we still have suspicious of this bacteria to carry a carbapenemase, we perform the MALDI-TOF imipenem hydrolysis assay to measure if there is a carbapenemase activity or not. If the result is positive, we then perform, this is a bit is wrong, but we then perform the CARBA-R GeneXpert. If it is negative and we still have suspicious, we perform whole genome sequencing, and if it is positive, we just gave the result as it is. If it is MALDI-TOF negative and CARBA positive, we just also perform whole genome sequences to get sure that there are no variants or something like that we cannot detect. I'm going to change a little bit of approach now. I'm going to speak about the quantitative MALDI profile. That is a completely different approach. In this case, what we measure is the growth of bacteria and the presence of an antibiotic. It's very similar to what we perform in microdilution susceptibility testing. But instead of measuring the growth after 16-24 hours, we perform the measuring of the growth after 4 hours, and the measurement is done in the MALDI-TOF. So when bacteria is susceptible to the antibiotic, it is unable to grow. So you, after this 4 hours of incubation, you won't see any mass peaks in the spectrum. But if the bacteria is resistant to the antibiotic, you will observe some mass peaks because bacteria is growing and it expresses the proteins, and you will find more or less accurate spectra, and not as good as it would be if you wait this 16 hours. But there is a very this is an accurate spectra. So you compare this spectra of bacteria with and without the antibiotic, and this is what we can find. What you can observe in Figure 1. This is a pseudo-Hell view, so you can just imagine you are in the top of the spectra. But it is clear that when bacteria is unable to grow, you won't find any peaks. So that is the main purpose of the approach. Evgeny Velevich made a lot of research in this area, and he improved the method up to now that you can incubate the bacteria with the antibiotic directly in the spot, and you make the separation of the broth from the microbial cells just by touching the micro droplets with a filter paper. and afterwards you spot the matrix in different spots, and you perform the measurement. So you can see that if there are no mass peaks, no growth, the MALDI biotyper is unable to perform any identification. So the score will be under 1.7. But if there are mass peaks, the software will give you an identification result. So that is in line with being non-susceptible. Another different approach is the peak profiling, which is in line with what Belen told you before in her talk. You measure the bacterial proteome, and you just compare between susceptible and resistant isolates, and you focus on finding some peaks that are different in both populations. So here the mass peak at 2,415 was indicative of the presence of the AGR positive methicillin resistant Staphylococcus aureus. So you can identify it by using this mass peak. This other approach is the detection of colistin resistance, and it is very interesting because the research workflow really may take days to perform the lipid extraction, as it is based on measuring the lipid A in bacteria and see if this lipid is modified or not. In the routine workflow, you would perform standard susceptibility testing, and if you find that the bacteria is resistant, you afterwards perform some PCR or some dipicolinic acid inhibition, but it usually takes 1 to 2 days at least. With this MALDI approach, the MALDI test, you can know if bacteria has the intact lipid A, or if bacteria has this lipid modified. So this is the mass peak of lipid A in E. coli without any modifications. And you have these two structures represented by this spectra. And you may also you can differentiate between the chromosomic and the plasmidic resistant mechanism, because the modification they perform to the antibiotic to the lipid A is different. So you can detect colistin resistance, but as well you can differentiate if this is produced by the MSR gene or it's chromosomically. So it is very helpful. We have also another approach. In this case, Paul Griffin tried to detect Enterococcus faecium Bambe producing, measuring the difference in the mass peaks in the bacteria, and it gave good results. However, Brackman published this very interesting paper, in my opinion, which changes the things a little bit. So he analyzed Enterococcus faecium Bambe, and he realized that he had also this mass peak that Griffin talked about, that was ranging 5,000 Daltons. And he performed a real analysis of this mass peak with proteomics. So he realized this mass peak was a hyacinth that was a secretory protein of bacteria that was anyhow related to the resistance gene. So he also analyzed computationally different genomes that was in the gene bank, to check how was distributed this gene and Bambe gene and the hirasin protein. So he realized that there were not many Bambe Enterococcus faecium which really had this hirasin, and also the hirasin was present in other Enterococcus faecium which didn't carry this Bambe gene. So he already advised that the pure correlation among the presence of a mass peak and the resistance cannot be made. So we have to really analyze what is this mass peak, this biomarker that we are using to differentiate bacteria. In this line, Analao published this paper in which she tried to differentiate and tried to track a KPC producing Klebsiella pneumoniae by this mass peak at 11,000 Daltons, more or less. And she talked about this mass peak as related to the PKP-kill plasmid, which was present in KPC, or KPC was part of this plasmid. Several papers appear afterwards validating these procedures with, I think, sensitivities ranging from 85% to 100%. So they were pretty good, apparently. There is also an automated software for interpretation from Bruker in which you can make an identification, and afterwards, if they detect these mass peaks, they give you a warning message. So maybe you have this KPC. What we try to study in our group, because we have a national survey collection of isolates sequenced, fully sequenced with whole genome sequencing, and we also studied these isolates by MALDI, and we realized that there were a lot of KPC isolates that lacked this mass peak. So we analyzed the genomes, and we realized that this Beta-Lactam gene was not only present in the PKP-kill plasmid, but was also present in different plasmids, and was absent in other plasmids that carry the KPC gene. So this direct correlation cannot be made. So there are different plasmids carrying this gene, and we also try to computationally analyze the situation in Europe. So for that, we use the EUSCAPE collection of isolates that was published in Nature last year. And we realized that there were huge differences among countries, because in Italy, the rates were almost 90–100%, but for example in Portugal or Poland, almost none of the KPC producers have these mass peaks related to the Beta-Lactam gene. So this approach has high variability and a really low confidence. So you first have to analyze your genomes, your bacterial genomes, so you can assure that this biomarker you are observing in Malditel is really indicative of resistance, or maybe not. In this line, we try to perform a different analysis to see if just not the specific mass peaks could help in differentiating bacteria carrying carbopenimasas, but using the whole spectra could help us better. And we find that using machine learning algorithms and the whole proteome could really help in differentiating this bacteria. We have 100% of accurate identification of carbopenimasas, and 82% of the isolates were correctly classified in the type of carbopenimasa, OXA48, KPC, or NDM. We use the random forest algorithm and some basic specifics that we explain in this paper. Both papers are under review now. This is a random forest features importance, in which you can observe the importance the algorithm gives to each mass peak. So it is, in my opinion, it's a quite better approximation approach, because you consider the whole spectra and not just one or two peaks. All mass peaks may have a relative importance in the final algorithm, but you take more information into consideration. So the differentiation is better. I think from now on it will be key to use proteomics with machine learning algorithm. Caroline will talk about it later, but I think this will be really the future of MALDI-TOF, especially in antimicrobial resistance detection. The advantages is that the results of resistance are provided in minutes, and at the same time that the identification. So it is very helpful, because you don't have to perform a hydrolysis assay apart from the identification. You just take the same spectrum into consideration, you perform the identification, and then you apply the different algorithm for the detection of resistance. And it is, as it is automatically performed, it just takes minutes, so it is very easy. It is less influenced by the expression of certain proteins, so variability is clearly reduced. And the inter-center transferability is also guaranteed. And that's all. Thanks to everyone who's listening. And I also would like to say thank you to my baby girl, which I really spent less time than I would like to these recent days, especially with this COVID thing now. So, well, I like to put a picture of her here. So thank you.
So thank you very much, Marina. It was really, really interesting talk. I mean, antimicrobial resistance is really a kind of really urgent problems nowadays. So we have a few questions for you, in fact. So the first one is from Anne. Is it able to discriminate the different carbapenemas? I'm not clear on that.
Sorry, if I'm able to differentiate the different carbapenemas?
Yeah, exactly.
With my last algorithm, or?
Well, it's not specified, but this is.
Yeah, I don't know if I can put the presentation again, maybe. Or?
I think you can go back in your presentation.
Okay. Maybe it's easier. Yeah. So we. In this study, we perform an initial differentiation among carbapenemasa producers and non-producers. So we could clearly separate both populations with 100% of accuracy. So they were really different. And afterwards, in a second step, we just try to differentiate among the carbapenemasa producers and in the different classes. So we could clearly differentiate KPC from OXA48. So the global accuracy was 82%. We have to say that for OXA48, we have more like 95% or 98, if I can remember, of accuracy. But we have more OXA48 in our area. So the algorithm was a bit biased as we have more of these isolates than the other ones. But we would like to keep working in this to increase our collection of isolates, because this was a survey that we performed in Spain with like more than 400 of isolates. But it is true that in Spain we have a clearly higher prevalence of OXA48. So we are working on this, but it is really interesting and we have very good results.
Okay, we have a second question from Jack. Did your team evaluate the Bruker kits from gabapenimase or the cephalose as? Or do you have any feedback on their sensitivities?
I think you mean the software from, you have this typing module, which I don't have a lot of experience in it, but if you mean the one from the measuring of the hydrolysis, get into it. If you mean this one, yeah, I have certain experience. It works really nice. You have the Kaba kit and the Cephag kit, and then the automated software. Yeah, it works really good. You have very good results. I think sensitivity in this type of assay is really high. The inconvenience is maybe the price, because it is true that you have to buy the kit apart from your equipment. But it's similar to any other technique.
Okay, so in the meantime, we just received over questions. So we have one from Stefan. He just say, do you use MALDI-TOF for over application than bacterial identification or OXA48 meropenem hydrolysis in routine practice?
Yes, I use imipenem hydrolysis. I really prefer imipenem because it gets results faster, and I think it is more accurate. Because with meropenem, Jaroslav Habrak, that usually uses this approach, he uses a different matrix because the crystallization is better if you don't use the common thinamic matrix. That's the reason, because for that I'm using imipenem. But it is true that usually I perform in the first place an immunochromatography assay because it is simple and it is easier for technicians to use. But if we have any doubt, we perform the MALDI in the second place, and it is really very useful because we are increasing the amount of Klebsiella pneumoniae that are resistant to ertapenem or even meropenem and lack carbapenemase. So when you see just the susceptibility testing, conventional susceptibility testing, and you observe this carbapenem resistance, well, you get more confident if you perform a MALDI assay because you measure the real activity of the enzyme. So for me, it is quite helpful to implement control measures at hospital.
So in fact, we have all the questions. However, we need to stay on time on the agenda. So I would suggest that we will send you later the question that we just received for you, and then maybe you can answer it by emails or.
Yeah, of course. Yeah, I think I have my email in the first slide, so you can write me if you need anything.
Okay, so well, thank you very much, Marina, again for these really nice presentations. So we just reached our coffee break now. So we have a 5 minutes break for grab anything you want to grab for drink. So we will be back in 8 minutes. So see you there. So we're back now. So let's move on the schedule. So the next presentation will be the one from Caroline Weiss. So she's a PhD candidate at the ETH of Zurich, working on machine learning for healthcare in the group of Professor Carsten Bogbart. Our main focus lies on developing models predicting antimicrobial resistances from Malditel mass spectrometry data by exploiting domains such as topological data analysis, kernel methods, and representation learnings. So we just need to open your presentation, Caroline. Just a minute. Here it is. So Caroline, the floor is yours.
Yes, thank you to the organizers for organizing this symposium and for inviting me to speak here. I think I do not have power over the slides yet. No, okay. So so when I was preparing the slides, I decided to introduce the topic of machine learning on MALDI-TOF mass spectra a bit more generally, and to include some information on why applying machine learning to MALDI-TOF mass spectra makes sense, and talk a bit about the current state of the art of MALDI-TOF based machine learning, which we have summarized in a recent review. And only later I will introduce a machine learning method tailored to MALDI-TOF mass spectra, which we have developed called PIKE, the peak information kernel. And if time allows it, I will go into confidence estimates, but let's see. Okay, so why should we apply machine learning to MALDI-TOF mass spectra? Actually, Marina, at the end of her talk, already gave nice reasoning for that, but I will illustrate this as well on a short toy example. So if you have a data set structured like this, if you have different MALDI-TOF spectra, and some come from sensitive or susceptible samples and some come from resistant strains, why should you use machine learning to predict the antimicrobial resistance instead of looking for biomarkers in a univariate fashion? So if you look at the spectra, you might not see any differences, or if you would apply univariate testing, you would not find any significant biomarkers. But if you can apply machine learning algorithms to this data set, you will find patterns like these, where if you have, like, if any of the peaks in these three highlighted peaks is lower than the others, you suddenly get a signal that that you can use to predict resistant isolates. And also here, for example, there's an example of a group of peaks that you can use in a combined fashion to indicate susceptible samples. So machine learning is able to detect more complicated patterns across the spectra, and utilizes predictive features that are not individually necessarily statistically significant. So you get a more holistic view of the entire MALDI-TOF mass spectrum, and that is quite powerful to predict properties of the strains. So yeah, so as I mentioned, a colleague and me, my colleague Catherine Yüksel and I have written a review to determine the state of the art of MALDI-TOF based machine learning. And we looked into two properties that you can predict from MALDI-TOF spectra, namely the species and antimicrobial resistance. And to give a very concise summary of our findings, we found that practically all studies used out-of-the-box machine learning methods, so methods that were not specifically designed or adjusted to the data type of MALDI-TOF mass spectrum. The data sets used in these studies consisted of a few dozen to a few hundred isolates, and often studies would lack proper validation or validation of the results on holdout test sets or on completely external test sets. And just to give you an idea, from coming from a machine learning background, for a task like this, predicting antimicrobial resistance from these spectra, having about 500 samples gives you a rough idea of where we would consider to even that it starts making sense using machine learning for these tasks. So a lot of these studies used really, really small sample sizes. So what we could conclude with that is that MALDI-TOF based machine learning research is really in its infancy, and while gaining popularity and more papers are coming out, no significant strides in terms of machine learning research have been taken yet in the field. Just coming from the field of machine learning in computational biology and healthcare is actually quite disappointing, because machine learning healthcare has been getting so much attention lately, and the field is really soaring. And whenever I present my project at machine learning conferences, the topic is met with huge interest. So where does this disparity come from? The largest limitation is actually quite easy to identify, and it's the lack of publicly available data sets. So I'm aware of the massive repository and the data sets by Robert Koch Institute. But as far as we could find, often there are no resistance labels. So antimicrobial resistance prediction is not feasible. And also often these data sets are not in a standardized format, or the documentation would be unclear to someone who doesn't have a strong background in MALDI-TOF. So it's not exactly an easy entry for machine learning groups working on computational biology to get into this field and start looking into these problems. I do want to make a case that machine learning has great potential in the field of MALDI-TOF. So improving prediction through more complex and tailored models is obviously an integral part of the machine learning field. So this is very applicable to the task of MALDI-TOF based machine learning. Being able to give precise confidence estimates along with your prediction is also a part that is very well worked on, studied on in the field. Representation learning is also its own field in machine learning, and that's been used to mitigate batch effects in biological data sets, which again is very applicable to MALDI-TOF. So learning a representation of MALDI-TOF mass spectra that are independent from influences such as different instruments or growth media or slightly different lab protocols would be, I think, very useful. And the tools and methods are out there. That just haven't been applied yet, really. Also analyzing what pre-processing could be done to the mass spectra and how that influences the predictive results is also very interesting. Yeah, and the list goes on, really. So what I'll present briefly is a method that goes right into this direction that was accepted at ISMB last year, and that is based on kernels. And kernel functions in machine learning, if you're not familiar with them, just for the sake of this talk, think about them as a similarity function. So given two objects, the kernel will output a similarity score. And these kernels are developed for different types of data inputs. So for example, you can have a graph kernel that would take in two graphs and as input objects and output a score on how similar these graphs are. Same goes for text kernels, same goes for sequence kernels. And then there are these machine learning classification algorithms that are based on kernels, such as support vector machines and Gaussian processes. And in the algorithm, a kernel function is used as the central component of the algorithm to calculate similarities between samples. And the neat thing is you can exchange this kernel function. As long as it's a kernel function, you can plug it into your algorithm, and you have a machine learning classifier that's adjusted to your data type, for example. And this is exactly what we did in this work. We designed a kernel specific for MALDI-TOF mass spectra and plugged it into a kernel-based machine learning classifier. So we had the first really tailored to MALDI-TOF mass spectra machine learning classifier. So we designed PIKE, the peak information kernel. Just short how it works. So given two spectra, S and S prime, you have the M to Z values, Xi and Xi prime, and intensity values, lambda I and lambda I prime. The closed form, so just the kernel function of is displayed here. So the idea in PIKE is that every peak in S is compared to every peak in S prime. This is what this sum does. Then the intensity values of each peak pair are multiplied and weighted by this exponential function that was inspired by the heat diffusion equation. What this weighting does is that the further away two peaks are on their M to Z values, the lower their weight is in the comparison. And this allows us to calculate similarities based on peaks. Through this each pair comparison, we are able to assess interactions between peaks, and we only have one parameter, which is nice for machine learners that we need to optimize. And we've plugged in this PIKE kernel into a Gaussian process to have our method called GP-PIKE. Our data set that we worked on comes from the University Hospital in Basel. It was collected in the clinical routine in 2018. Includes 3 species, E. coli, Klebsiella pneumonia, and Staph aureus. And we used 3 antibiotic resistances as our labels. And you see here the properties of the data set. And we define a binary classification scenario. So we classify, is the spectra coming from a resistant strain or not? And these are our results for the 9 scenarios. So we compare to 2 other classifiers. First, logistic regression. Logistic regression is a linear classification algorithm, and it's very widely used in sort of the workhorse in the machine learning field. And a Gaussian process with a different kernel. So an RBF kernel, which is sort of the standard kernel that we use for most tasks, and our GP-PIKE. And as you can see, we achieve superior performance with GP-PIKE on all 9 scenarios. Quickly check the time. So we quickly looked into estimates, confidence estimates. So we would argue that assessing the confidence of your prediction, so if the classifier is sure of the output that it's giving you or not, is indispensable in this field of application. Because if you're thinking about a clinically applied resistance predictor, that the predictor will be given samples that it is not sure about, because a bacterial strain is introduced or comes up in the clinic that is not part of your training data set. So if a patient comes in with a strain from traveling, for example, and also changes occur in the strains over time. So you need to be able to assess the confidence of your model outputs. As the confidence, because we have a binary prediction scenario, if you use each class will be given a class probability. If you use the max class probability, you will get the probability of the class that is assigned. So the maximum class probability will be between 0.5 and 1, and that measure is what we used as confidence estimates. Very quickly, so what we did is we did this for logistic regression and for GP-PIKE, and we just looked at the distribution of how these maximum class probabilities are distributed among samples from within the training distribution. So we used the same species, and to sort of simulate out of training distribution, we used the samples from the other 2 species. So what you can see here is a prediction of Staph aureus and yeah, exactly. And in the out of distribution species are E. coli and Klebsiella. And what you can see is that within the training distribution, the maximum class probabilities are distributed like here in this first row. They're distributed sort of over the entire range of values, so between 0.5 and 1. But what we've observed when we give the classifier out of training distributions is that for the logistic regression, we get values very close to 1, which you can explain sort of with the linear decision boundary of the classifier. So if you have your training distribution, the classifier will learn like a separating plane to separate resistant from susceptible samples. But if you give it a sample that lies very far away, because it is out of the training distribution, it lies far away from the decision boundary. Therefore, the classifier will give it a high confidence estimate because it thinks this is like a sure thing. It's very confident about that. So this is actually not what the behavior that we want. So we would give falsely confident estimates. But what GPPike is doing is it gives the unknown samples a lower maximum class probability. So GPPike is able to recognize its own inability to classify these unknown samples. So it will give a class, but it will say, I'm not sure about this. So this is a very nice behavior. And to wrap up, so we've developed GPPike, a novel approach to antimicrobial resistance prediction from MALDI-TOF mass spectra. It is specifically designed, like our kernel pipe is specifically designed to MALDI-TOF mass spectra. And in combination with the Gaussian process, Pike outperforms methods traditionally used in MALDI-TOF-based machine learning, while being able to provide reliable uncertainty estimates for its predictions. Yeah, so I hope these results from the prediction task and also the systematic review have demonstrated how exciting the field is of MALDI-TOF-based machine learning and of its potential to improve antimicrobial resistance prediction. So if you have any ideas how to advance the collaboration between machine learners and MALDI-TOF researchers, or if you are interested in collaboration, please don't hesitate to contact me. And finally, I would like to thank my co-authors, Catherine, Max, Bastian, Carsten, Aline, and Adrian, and the whole group of MNCB for their support. And with that, I would like to conclude. If you have any questions, please don't hesitate to contact me via email. Thank you.
So thank you very much, Caroline, for this really nice presentation. Honestly, I think machine learning is kind of really the future in microbiology in general. I'm not a specialist in machine learning, of course, but I really think it would just help to move a bit more in clinics. So of course, if you have questions, please let us know. However, we have a comments from Ray. So we're in complete agreement with your approach. We have an issue when moving into half mass peak analysis around 50,000 to 150,000 kilodalton. When you're incompletely resolving glycovariant on top of isotopic mass effect on the peaks, we have to adapt from peak height to peak areas as measure comparator intensity. The problem is baseline definition. Have you any experience in this?
Okay, so this is a very specific question.
Pretty long question, in fact.
I usually get the spectra in the mass range of 2,000 to 20,000 kilodalton, and this is the spectral range I can say something about. Yeah, it kind of asks for further discussion. So if the person could just email me and give a bit more detail, I would be grateful for that.
I think anyway you already have email of each overs, like Ray's talking tomorrow, so you have some discussion about it more privately about this. But it's a really interesting question. Well, remark questions. Okay, so apparently there are no more questions for now. so we suggest we move. Like this, we're just still on time on the schedule. So thank you very much, Caroline, for your presentation. It was really, really nice.
Thank you.
So let's go back to our main PowerPoint. So our next speaker will be Dr. Jörg Rau. He's a food chemist and is working as a head of department for food of animal origin at the Chemical and Veterinary Analysis Agency of Stuttgart. So as a part of the official food control in the state of Baden-Württemberg in Germany. So his area of experience includes the use of further development of identifying spectroscopy, like Maldi-TOF mass spectrometry for food testing and veterinary diagnostics. So Jörg, the floor is yours now.
Okay, you can hear me?
Yes.
Okay, you are wonderful. Thank you, Maureen. In my presentation, I would like to introduce MALDI-UP, the MALDI user platform, not only for microbes to you. This platform is located at the internet address you see in this button here. Our background, the Chemical and Veterinary Analysis Agency Stuttgart is a regional state laboratory located in Baden-Württemberg in the southwest of Germany. As Maureen said, we are dealing with food control, food microbiology, and any chemical health risk like pesticides or food contact materials. Our second part is the analysis of samples from all kinds of animals, especially zoonotic agents and causing agents of any animal disease. Identifying spectroscopy is used for microbes since 2004. With firstly introduced by infrared spectroscopy, and MALDI is added 2013, also first for the usual application, microbiology. For microbes, we use a workflow from MALDI-TOF mass spectrometry to infrared spectroscopy. The MALDI-TOF MS process is a common, as shown by Belen in the first talk of today. Bacteria were extracted or smeared directly on a steel plate, overlaid by matrix to obtain a molecular protein fingerprint in the mass spectra. For infrared spectroscopy, the workflow is similar. Starting with a pure culture on solid media, dried bacterial films gives an infrared spectrum, a fingerprint of bacterial components which can be compared with reference spectra. The identification is done by assignment to reference entries in a database too. We use MALDI for the species level and infrared spectroscopy for subspecies, for analysis of identity or for contamination routes. Since 5 years, we expand the MALDI applications to other fields, like the animal species identification of milk or meat. Similar to microbes, we use concrete biomolecules of skeletal muscle, for example, myoglobin. Myoglobin has mass charge ratios in our usual window. Different animals shows different Dalton values for this major protein, as here shown for wild boar and raw deer. The sample preparation is easy to handle too. Starting with frozen meat, we drill a pin-sized subsample. Then the tissue is mechanically abraded in the usual organic solvent mixture using pistil and zirconia beads together. And the further protocol steps are completely the same as you know from microbes. Preparing the steel plate, adding matrix, and measurement with the normal device settings in the MALDI system. A description of this protocol and some other protocols is given in on the MALDI-UP page. For the final identification of the sample spectra of some meat, we use a special own database. Our meat database comprises today more than 260 species and shows partly a very good resolution between them. A formal validation on basis of more than 1,000 individual meat samples is done too. A smaller, but also often used application was created for cheese. Mozzarella from buffaloes or cow's milk and feta from sheep or goat and cow are connected frequently with food fraud problems in restaurant samples. MALDI helps us here to get a valid result for the main animal species in 50 minutes. For any application, the database is the key. Sources of databases are the manufacturer or other commercial companies. Also, several free downloads can be found. For us, the main source is own activity and the exchange with other MALDI users. We and our colleagues in Baden-Württemberg from our sister institutes in Freiburg and Karlsruhe created more than 1,600 reference entries together for several applications: microbes, meat, but also seafood, especially fish, insects, fungi, and so on. To utilize this treasure of reference spectra more effectively, an exchange with others will be helpful. There are some basics for any exchange. You need confidence on the work of your colleagues. A consequent, transparent presentation of metadata helps here. And in the timeline, you have to care of your data. It's really necessary to give a transparent status of the data. To support transparency for the direct exchange, we create an easy, non-commercial who has what catalog called MALDI-UP. In MALDI-UP, there's no direct download possible. It's really only the information who has done what and who wants to exchange with everybody. In MALDI-UP, you found an Excel file for download, and in several sheets deliver basic information for now more than 2,300 reference spectra and more than 5,000 validation spectra. Informations are given like the creator or owner of the spectra, the sample preparation method used for the multispectrum, or verification information for the material used, because we need this confidence. In the same way, data for single spectra for validation purpose is presented. This catalog should support the bilateral user-to-user exchange. Now I want to show you two examples from veterinary medicine, shows you the benefit of a fast exchange. The first example is from my colleague Tobias Eisenberg from the Hessian State Laboratory. Last year, 2020, we observed a disease of blue tits in Germany. A few days after isolation of the first suspicious bacteria, the colleagues of Gießen create some reference spectra for MALDI-TOF mass spectrometry. They listed these reference spectra in MALDI-UP a day later, and more than 12 other interested MALDI users have updated their databases with this reference spectra for so-called Sotonella ornithocola. It's a bacterium not represented in the commercial databases yet. This new reference enables a direct impact of the diagnostic possibilities of the laboratories. In the second example, the sharing of single spectra between 6 labs enables the validation of the MALDI method of a fresh new species. Corynebacterium sylvaticum, a close relative of Corynebacterium diphtheriae, is causing onion-shaped abscesses of wild boars. For the validation, we collect spectra from several states in Germany and Austria. In MALDI-UP, now we see contributions of more than 30 institutions from Germany and other countries. So MALDI-TOF mass spectrometry is an integral part of our everyday workflow in microbiology and food control. Active database creation is successful and pays back. The active exchange helps us to reduce the workload by implementation of new parameters and in the validation process. And in future, further expansion of databases will occur. Additional application will be more common also for other MALDI users like plant, fungi, seafood, insects, or whatever. For these activities, new databases and validation processes are necessary. So exchange is helpful and succeeds if basic standards are given. In Germany, we have now a work group dealing with such standards for the food control sector, located in the Federal Office of Consumer Protection and Food Safety in Berlin. So we look forward to exchange with you, if you want. Further information, links, references, some hints to events, contacts, and so on are listed on the MALDI-UP homepage. So thank you for your attention.
So thank you very much, Jörg, for this really nice presentation. So, so far we do not have a question, but I have one for you. So you just present that your platform for more, let's say, open science and more open collaboration. So I was wondering, like in the presentation before, Caroline say, for example, that there's not a lot of open access for Spectra for resistances. Is currently your platform have, for example, bacteria the label for antimicrobial resistances?
Up to now, no, not in some spectra, yes, but not in common, because in the first step we think about species identification. But it will be, because it's a very easy Excel-based list, it's not very complicated to add several information if there was a user who wanted. But the benefit for the presentator of the information should come back. So it's a win-win situation. So it's not only for give away anything on information, we will want to get a recycling of our work with this catalog.
Of course. So in the meantime, we get questions from Kathleen. How about fish meat? Did something
fish
about
meat,
it?
Yes. We have, I know that there are several databases of fish, and in MALDI fish spectra are integrated, and you have to download and see what the colleagues have presented. We in CBU Stuttgart have no fish species there, but the colleagues in Freiburg and Karlsruhe and some other have fish spectra presented there.
Okay, and I was also thinking, is your so when you have access to the database, for example, on the Malditel, do you have like automatic update, or you have to make the update by yourself?
You make the updates by yourself. It's not a database you get anything from everyone. It's a real bilateral exchange. You get only some. If you want only veterinary microbiology samples, you can focus on that, and you don't have to take the meat samples together. It's really little packages.
Okay, so I do not have more questions for you, and apparently there's no more question also for you from the chat. So I suggest that we move on the schedule. So thank you again, Jörg, for the presentation. It was really interesting, and I think the veterinary part of the Malditel has a really weak representation on symposium. So thank you to be here. I'm really glad that you make this presentation. So thank you.
Bye.
Bye. So our next speakers and our keynote speakers also is Alex van Belkom, Professor Alex van Belkom. So he's from Biomarkers company. So he's a microbiologist with an extensive academic background. So he has PhD degrees from the University of Leiden and the Erasmus University of Rotterdam, both in the Netherlands. He joined Biomarkers over 10 years ago and have several posts within the company. So he has been involved in the development of the Vitek mass spectrometers and accompanying databases. And he's still currently involved in critical assessments of the performance of the technology. So also quite important to highlight, he has published close to more than 100 PubMed cited papers, and it has a Google Scholar defined H index for 100. So, please, Alex, the floor is yours.
Good morning. I hope you can all hear me. If you look at the picture on the slide and you see this face now, then you will realize what 10 years industry do to you. Aging is not easy. The other thing I want to start complaining about is what Maureen said, and I want to thank her very much for the invitation and the fact that everything is running smoothly up until now. But there's still one big issue that I have, and that normally if you're a keynote speaker, you're at the beginning of a session. Usually keynote speakers are older people that know a lot about a lot of things, who know little about a lot of things, and introduce a topic, and then afterwards the real expert come and explain what the keynote speaker intended to say. In this particular case, the order is reversed. So everything has been told already, and now it's up to me to say something that has not been said before, and that is complicated, but I will give it a try. And I hope you bear with me that some of the slides will contain information that was already alluded to by the other speakers. But I think it's also good to repeat certain things and to make sure that the right message sinks in. And this is again where I need to issue a warning. I do not agree with everything that the previous speakers told us, and I might get back to some of the items that were discussed and provided as given facts, but which I think are not yet there. But then again, we'll see where that ends up. So in order to sketch the scenario a little bit, and this is a typical keynote #1 slide, is what are we working on, and where does our field of expertise stand at this stage? And in this case, we're all about diagnosing infections because, you know, like the previous speakers all alluded to, identification of bacteria is important, but identification of a pathogenic bacterium is even more important, especially if you have a fever. A patient with fever and or a big abscess, and you want to treat that person with an antimicrobial, the normal anti-infectious agents. For that, it is very useful if one, you know the bacterial species name, because that already tells you something about the treatability of an infection and what category or what large category of antimicrobials you need to look into. And the other thing is this antimicrobial susceptibility profile. If you know what the infectious agent is susceptible to, then you can treat a patient in a targeted fashion. And what you very frequently see still is that patients with infections are treated in a generic way with a broad spectrum antimicrobial that is not targeted to the agent. So if you look at this somewhat complicated but not too complicated scheme, then there's a list of technical means. So you can look at host factors and see whether these are elevated or declined and indicative of an infection. But mostly people use classical microbiological technologies, which are usually slow, which are well accepted, which have been around for more than a century now, since Pasteur and Koch, which are very much appreciated and which are slowly being supplemented and replaced by other technologies. Now I'll get back to that, but molecular diagnostics is one, and mass spectrometry, obviously, as you see in the second box here, is another example of a recent technology that improves this patient management approach. Of course, then there's the antimicrobial susceptibility testing, for which a wide variety of methods are available. And although some of the previous speakers talked about AST, antimicrobial susceptibility testing by mass spec, I'll get back to that with some of my own problems in that particular field. But still, this is the most important part. What is the antimicrobial susceptibility profile of the organism, and how can we arrive at that conclusion as quickly and as reliably, which is also a very important aspect, as possible. And then in the end, there's also resistance levels. Is this a highly resistant organism, or is it borderline resistance? These are also clinically important questions, because that will also be defining factors in the decision making with respect to what antimicrobial to use. And I'm not going to repeat for all of you that antimicrobial resistance is important, that it is a global problem, that there's millions of people who are going to die from infections due to resistant organisms over the decades to come. I presume that you know that, and that you're aware of that let's say clinical problem. So what about classical microbiology then? And you've seen it in many of the slides of the previous speakers. Culture is very, very important. And this is also immediately one of the drawbacks that has not been addressed yet by any of the speakers. You need, for mass spectrometry, for MALDI-TOF analysis, you need a certain biomass. You need 10,000 organisms in order to generate an interpretable spectrum. There are technologies that are suited for single cell mass spectrometry, but these use equipment and technologies and protocols and procedures which are to date not amenable and not compatible with clinical care. It should be quick, which should be cheap, which should be high throughput, and these are the three criteria that are missing with this new category of mass spectrometry applications. I will get back to that at the end of the story. But this is important. You need to realize that before your MALDI analysis, in most cases, you need to biologically amplify the organism that you wanna identify and characterize with respect to its antimicrobial susceptibility level. And some of the culture technologies are already doing that in part. There are selective screening media that do not only identify organisms by the color of the colonies, but that they also only reveal the growth of organisms that, like in this case, are resistant to colistin. So there you have a pre-enrichment of the organisms that you want to look into in more detail with your mass spectrometry approach. And there I also have to emphasize that the plates with the organisms on it should not be thrown into the mass spectrometer without any thought. You need to look at these plates, and also a visual interpretation of the plates can help you to select or to at least include all of the organisms that are important for the subsequent mass spectrometry analysis. If you take this pre-poured medium, this culture plate, then you can see with the naked eye that there's at least two species, Staph aureus and Pseudomonas aeruginosa. And if you look at Pseudomonas aeruginosa and you can recognize these by their colony morphology, then you will also see that there are different colony variants. And you need to take those into account because mucoid or small colony variants of versions of Pseudomonas aeruginosa may have a clinical impact. This can all be automated. Pictures can be taken. Microbiologists can do this from the confinement of their homes, which is particularly important in these troubled COVID times. But again, before you start doing your mass spectrometry, do not ignore the quality of the culture and the relevance of looking at the culture in order to sort of a pre-assessment of what you want to measure in your mass spectrometer. Another thing I want to highlight, and which has not been addressed by the previous speakers, is that mass spectrometry is not the holy grail. It is a fantastic, don't misunderstand me, it's a fantastic method for bacterial identification and further characterization, but there are things that are not yet realized. The equipment may be bulky and big. The fact is that it's not an open access system where you can put in at any moment that you want your next specimen or your next clinical specimen is a drawback. And some technologies are better at certain aspects of this clinical diagnostics than mass spectrometry is. And we published something on NMR as a tool, which is not a very good example of a technology that's going to replace mass spectrometry in the lab anytime soon. But it's just to illustrate that in some cases, it's also good not to look at the organisms themselves, but at the metabolites that they produce during growth, because that may be a little bit more sensitive than looking at cell numbers. And it may also lead to signals that are generated earlier in the growth cycle of organisms. But the only thing I want to specifically state here is that Mass spec is in the lab, it's well accepted, it's been embraced by the microbiological and the clinical community, but there are parallel developments there that you need to continue looking at. And the two most important ones there, and I'm not going to detail that very much today, there may be another occasion for that, but you are all aware of the fact that PCR technology or other nucleic acid amplification technologies can be used for detection and identification of bugs from clinical specimens. And in many of these cases, these tests are so sensitive that you don't need that pre-culture on an agar plate or in a liquid culture medium, which is very important to realize, because culture takes between 6 to 72 or sometimes hours and sometimes weeks if you talk Mycobacterium tuberculosis. And PCRs can be done in 20 minutes or 40 minutes or 50 minutes within a very brief time span and very rapidly. And then next to that very competitive set of new tools, the molecular tools for microbial identification, but also for the detection of resistance genes, there is next generation sequencing, which using the Oxford nanopore technology, for instance, can also be done in an hour with numbers of DNA molecules that are relatively low. So with a higher sensitivity than mass spec has at this day and age. Again, PCR is more expensive than mass spec. Next gen sequencing is not yet ripe, mature enough in order to be introduced in routine clinical testing. It takes too much time and it's still expensive, but these are things that are evolving very rapidly. The timing of next-gen sequencing is on a steep decline and the costs is are on a similar decline. But still, this is not the talk of today. This is about mass spec and the fact that directly from a clinical specimen plated on a selective growth medium, you can identify a species. And I want to highlight two negative aspects of the technology here, because you are all aware of the fact that has been alluded to by the previous speakers, that you need your biological material, you need some chemical to facilitate ionization, and then you need to accelerate the ions. And that is problem number one, because that takes a relatively long tube, a meter minimum, more or less, which means that the equipment is going to be bigger and it's going to stay bigger than the average PCR machine. And in clinical microbiology, laboratory space is expensive, so the smaller the equipment, the more appreciated that is. But this is mass spec, and there's things that you can do with this acceleration tube. You can curve it, and you can do other things, but it will stay pretty bulky. The other thing is the fact that this all happens in a vacuum, and for a vacuum you need pumps, and pumps are heavy and noisy. You can isolate that pretty well, but you know, this is still something that is not very compatible with a friendly laboratory life. And the other thing with vacuum is that they are sensitive to perturbation. So if you put in your sample, then you disturb the vacuum, which has let's say practical implications. You're not going to put a new sample plate into the machine every two or three minutes, because that's physically complicated. But still, this again, I'm going to quit very quickly. This is where we stand. We have huge databases with thousands of organisms that all have their characteristic their species characteristic spectra and profiles. And within a species, you can also look at the peaks that are conserved, but you will also see that in some cases there are peaks that are not species specific, but isolate specific. So hence the typing application that has been addressed by Belen earlier this morning, and that will be discussed by my colleague Kathleen tomorrow in more bioinformatics detail. So I'll skip that for today and just put one more slide on the value of the databases. We've briefly discussed the technology, which is there and which is, like I said, mature and very well established. The databases are the other aspect of clinical diagnostics that are extremely important. One has to take care that all the variants of a certain species are well enough represented in order to be recognized as being part of the species. So global variability, a global strain collection at the basis of your spectral database is absolutely essential. Strains included from different sources. Jürgen Rau just talked about the veterinary part and the food part. These type of strains need to be included because they may be phenotypically different, they may show different spectra, but looking at the overall peak complexity, they clearly belong to a certain species. So doing a population-based development of your database, where you have at least a decent number of strains per species, is very important. And you'll appreciate that in our mass spec database, there's hundreds of strains of E. coli and hundreds of strains of Staph aureus and hundreds of strains of all of the, let's say, the better known pathogens. The other thing is technical variability. You need to culture your bugs from your population-based approach on different culture media in order to ascertain that certain peaks are not only produced, are only produced or not on a given culture media. And you need to play with operators and systems to make sure that if you use mass spectrometer A or B from the same manufacturer, that they perform in a similar fashion. And incubation timing is obviously important. If you do overnight cultures on a solid medium, or you do 2 hours exponential cultures in the liquid medium, then obviously this may generate different mass spectra. And also representative collections of spectra means that for a given species, we set up a sort of a bottom line of about 40,000 spectra in order to have a decent database allowing for the adequate detection of all isolates from within a species. And then, of course, you need algorithms. I'm not going to burn my fingers on that because I know little about that, but you know, the interpretation, the comparative analysis of the peak patterns is absolutely key to the appropriate interpretation. Then another thing that I want to point out very clearly, Vitek MS or the instrument that our competitor Bruker generates is there for daily routine needs. So it should be as simple as possible. It should allow high throughput. It should be reproducible. It should be consistent. It should be sturdy from a clinical perspective. And my friend Chand Wattal from India, he published a review study a couple of years ago where they analyzed about 10,000 different isolates in a year. So mark my word, they did 10,000 identifications in a year, which is more than 30, 40 on a daily basis in a private specialty care hospital in India. And they were able to show that about 96% of the bugs were correctly identified, which is very good. There are low discrimination species within the same genus. If you have Shigella and E. coli, there's a persisting problem there. There's obviously bugs that are not identified, which are very interested for the manufacturers, for Bruker and for ourselves, because these may be species that are not yet sufficiently represented or maybe even absent in your database. So including that is very important. And then of course there's always, because this is biology, there's always misidentifications because of mismanagement of a sample, because somebody forgot to put the matrix on a specimen, or maybe too little material was put on a plaque. So there are things that can still go wrong. But well, look at the worldwide COVID-19 vaccination campaigns, and you'll realize that we're just humans, and anything we do goes wrong to a certain extent. So this is important to realize that this should be easy. And there's this standard slide on picking a colony, putting it manually on the slide, and then you add the matrix and load it into your mass spectrometer. Even in that field, innovation is still an option. And I want to emphasize that what all of the previous speakers told you today is that there is a given protocol for the use of mass spec in clinical testing, but if you use your brain and you use your hands, then still innovation is around the corner. And we've developed a very simple tool that will allow you to, by picking a nib, picking up a microorganism, smearing it on the plate, throwing away the nib and start with the next one, to even facilitate a procedure that is manually already very simple to something that is even more simple. And this method, and we validated that in several customer laboratories, are 20 to 40% faster. And that's probably even more important. The deposit, the bacterial deposit on the slide has better uniformity, so you get more reproducible spectra, and with that, enhanced laboratory efficiency. Still, this is what you do with colonies on a plate. There's the colony, you see it, you pick it up, and you smear it on the slide, and then you can do all of the things with the complicated, the difficult species, again, as discussed by Belen this morning, but also with yeasts and molds. Two other specimens deserve somewhat more particular attention: positive blood cultures. This is one of the few clinical liquid, let's say, specimens that contain enough bacteria per ml to not have to be subjected to a culture post the liquid culture positivity on a solid media in order to generate these colonies and do this biological amplification. And the same is true with urine samples. Also, in case of urinary tract infections, the concentration of infectious organisms in the urine can be up to 10 to the 5, 10.5, 10.6 colony forming units per ml. So there you can directly take some of the biomaterial, spin it, lyse it, to do some preparations, and put the bacteria without cultivation on a solid culture media onto the mass spec slide. Important to realize that these are samples that are available, become available in the lab, and are important in let's say, providing a more direct diagnostic impact of MALDI-TOF MS. I talked about that difference between MALDI-TOF MS with the prerequisite of culture and sequencing or PCR as more direct on sample technologies. Direct on sample MALDI-TOF MS can be done with positive blood cultures and urinary tract infections or urine from patients with UTIs. And this has a very clear effect on the clinical impact. Already in 2013, papers appeared where people demonstrated that the shortening of the diagnostic period by mass spectrometry had a very clear effect on patient care. Again, I will not be discussing that in much detail, and I'll end this part of the talk with a brief illustration on what a positive urine sample looks like. If you spin down 1 ml of a positive urine sample, you'll see the red spectrum here in the middle with peaks that are generated by the infectious organism, and you can clearly see the overlap between the peaks in the clinical example with that in a E. coli calibrant sample where you see identical peak positioning, and where the species nature of E. coli is absolutely clear. It also has to be realized that if you do direct measurements in clinical materials, you'll also be picking up host molecules. And in the bottom sample, there is a negative urine, and the peaks that you see there are small molecules, small proteinaceous molecules called defensins, which are natural, I would nearly say contaminants, but which are naturally present in healthy urine. So you need to deduce these from the spectra of the clinical specimens. Another thing that bioinformaticians can take care of very well by modifying the algorithm a little bit. Two things that also have not been mentioned this morning: in several clinical materials mixed species assessment is an issue, so especially in the liquid ones that I mentioned before. If you look at positive blood cultures, then between 5 and 10% of these blood cultures are mixed positive. So there are multiple species swimming around in the same bottle of liquid culture media. And also in case of urinary tract infections, those can be mixed in nature. And my colleague Pierre Mahé and his bioinformatician colleagues published a paper a couple of years ago on, let's say, looking at spectra and trying to figure out whether the spectrum that you are analyzing is homogeneous and deriving from a single species specimen, or whether there's a mix of two different spectra simultaneously present in the overall spectrum, deriving from a mixed liquid culture, for instance, one of these blood cultures. And this is doable. The only thing is that that the ratio between one and the other species needs to be at max 10 to 1. If it then goes like 11 or 12 or 20 to 1, then the bigger species, the richer species will sort of overpower the minor one, and you won't be able to retrieve the peaks for that particular species. So it can be done, but you need to realize that there is a molarity or a relative amount that needs to be taken into account when you look at it. The other thing, again, like an example of what may be to follow, and the direction that many people are thinking now, is the analysis of microbiomes. Microbiomes are very important. We have different organisms, microbial organisms, on our skin, in our mouth, in our intestines, and the balance of the different microorganisms is important and can be a marker of health or predisposing towards disease. And with mass spec and looking at the complexity of peaks, you cannot identify all the species in a fecal transplant, for instance. Like I said, the ratio between the different species is not sufficient in order to really identify at the species level. But what you can do is, if you take a fecal specimen and you put it, you have to clean it up a little bit, but you put it directly on your mass spec slide, you'll get sort of arbitrary peak patterns. And these peak patterns are, let's say, indicative of floral complexity. So the more different organisms you have in your fecal specimen, the more complex the pattern will be. If you're close to Clostridium difficile associated phenomenon, then the richness of the flora may go down and the peak pattern will simplify. So in that way, you can sort of distinguish between a healthy, complex flora state as measured by many peaks in your spectra, and if that simplifies and you only see a few peaks, then you know that there is dysbiosis, and that this person who provided the sample is either already ill or most likely to become ill very soon. And this type of screening technologies, where you do not identify species, but where you look at the complexity of resident flora, might be interesting for, let's say, health assessment analysis in a somewhat generic manner. And I'm sure that if you think about this approach, you can come up with many other examples. If people are treated with antimicrobials, their flora, their gastrointestinal flora, for instance, becomes heavily perturbed. That's why some antibiotics also cause diarrhea. So there is a simplification of the flora. And in order to cure completely, it would be good if the flora sort of re-establishes itself in its complexity. And this is something that you could potentially easily measure with this type of mass spectrometry technologies. And there you can look at the species, but you can also do metabolomic analysis using mass spectrometry. And we've been able to show in mice, for instance, that both at the microbiome level and at the metabolome level, if you treat bacteria with antimicrobials, then the complexity goes down. If you take away the antimicrobials, in most cases, but not all, the flora sort of rebalances again, and also the metabolome becomes more complex and more alike the pre-antimicrobial treatment metabolome. So these are things that are important from a clinical perspective, but I'm sure you know that mass spectrometry can also be used for meat analysis, as Jörg told you before, but also polymers, beer, drugs, to look at the originality of olive oil or caviar, and also the identification of mosquito sticks, nematodes, fish can be done with mass spec. So there's a lot of things outside clinical microbiology that is moving. This is where I want to come back to, let's say, the second part of clinical diagnostics, the antimicrobial susceptibility profiling. I'm not going to repeat the definition. I'm not going to explain because that has been done before by Dr. Oviano, the different mechanisms that you can use in mass spec to detect resistance factors. That there's even, like in this particular slide, methods where if you look at solutions with increasing amounts of antimicrobials with a standard amount of bacteria, that you can see whether something is growing in like the first dilutions and nothing is growing in the 2 final dilutions where most of the antimicrobials is present. You can even do a quantitative antimicrobial susceptibility assessment. And this is important because, like I said before, higher levels of resistance may require a somewhat more aggressive antimicrobial therapy. So quantitative AST by MALDI-TOF is possible, but this is where I want to put like another two question marks. First, Marina talked about the antibiotic hydrolysis this morning. One of the antimicrobials or categories of antimicrobials that are also hydrolyzed by enzymes are the aminoglycosides. And maybe Marina has to correct me and I'm wrong, but I do not recall any paper where mass spec was successfully used for hydrolysis analysis of aminoglycosides in a reliable, reproducible, high throughput manner. And again, so this entire antimicrobial hydrolysis method, it's a very nice method, technically speaking, but it is at this stage hardly suited for high throughput application in clinical settings. If you have 1 or 2 multi-drug resistance organisms in a week, then you may be able to do this. But if you really want to do this high throughput, then it's difficult. And the other 2 problems with mass spec and AST at this stage, and I'm sure somebody will find a solution for that in the years to come, is that currently most of the assays have to be serial. In most of the automated AST instruments, at any time that you want, you can plug in another card or another cassette with another organism with hundreds of antimicrobials, and then the measurements will begin immediately. In mass spec, you will have to wait till assay 1 is done, and then you can continue with the next, and that may take some time and effort. Like Marina already said this morning, it is still largely manual and difficult to automate, so that's another problem. And the biggest problem for me is the fact that in the current automated susceptibility testing systems, and there's 4 or 5 on the market, you can look at 1 bug in combination with immediately 20, 30, 40 antimicrobials in different dilutions, and you get the result altogether. If you want to do this in the mass spec, you have to use different formats, you have to use different slides, and you have to do that serially, which may take more time. And again, it's largely manual. So this is a problem, but I'm sure that over the years to come, with data-driven infection management becoming more and more popular, this will surely change and become more effective. And this is where I was slightly disappointed with the between brackets with the talk by Caroline Weiss, because if I'm well informed, they did something alternative with machine learning procedures as well, which to me is going to be paradigm changing over the years to come. The fact that they published it in Bioarchives, where they showed that if you use your MALDI-TOF spectra in combination with phenotypic antimicrobial susceptibility data for the same strains, and you merge that, and they developed a method they call drying something, where they essentially merged the resistance data with the spectra, and came to novel insights with respect to the association between a spectrum and maybe hard to identify by the eye markers in those spectra associated with a classical antibiogram. That to me is going to be really paradigm changing. If you use a large publicly available data set combining mass spectra and resistance information, their claim is that they could predict reliably between 12 and 27 hours earlier than with classical approaches a MALDI-based antibiogram. And I'm sure this is the first and certainly not the last paper on this subject. And I'm looking forward to this particular type of analysis specifically, because if you have all data on patient parameters, on microbiological parameters, on technical parameters, sequencing, MALDI-TOF, and you put that all together into a model that's going to predict whether a bug is virulent, whether it's resistant, whether it's epidemic, based on only a mass spectrum, then that's going to be a spectacular improvement of the mass spec approaches. Finally, and very quickly, test development processes are complicated. I'm working in a company, and some of you in the audience are probably as well, and we all know that development of a product is very complicated. So just claiming that you can measure hydrolysis of an antimicrobial with mass spec is not enough to develop a product and to develop a tool that can be used in clinical microbiology. So you have to be very, very, very careful. You have to combine business decisions with scientific decisions. You have to look at feasibility. You have to look at test design. You have to verify the test, and you have to do validation studies. And with all of that, you have to do risk analysis. You have to develop protocols. You have to do health economy studies, which is becoming more and more important in modern medicine. And finally, you will have to do the registration at the CE level or with the American FDA. So don't underestimate the development of a new tool and a new protocol. And this is also why I'm not going to talk about new and more spectacular mass spec methods. My colleague Jean-Philippe Charrier, he published a paper a couple of years ago in which he showed that with new methods of mass spectrometry, so not the MALDI method, but other ionization-driven methodologies, you can, in single assays, define whether a bug is species A, B, C, or D, whether it's resistant to certain antimicrobials, and whether it's virulent, and also what its epidemiological type is, based on peptide mapping and peptide profiling. I'm not going to tell much about this. If you want to read about it, I'm sure that there's many other papers besides our own, but the only thing I want to say here is that in this particular case, this entire protocol for product development has not been developed, and so FDA approved products in this range do not exist. But this will come. Again, equipment needs to become a little cheaper. The open access sample accessibility of these systems needs to be improved, because now you can only do one sample and then do the next one 45 minutes later, which is incompatible with clinical microbiology requirements. But this is going to come, and this is going to again change the field of clinical microbiology. Some very rapid, because I see that my time is over, concluding remarks. Classical microbiology will not soon cease to be. A mass spectrometer is still expensive, and in developing economies, this is still a bottleneck and a hurdle. You also need significant amounts of electricity for your mass spectrometer, and as we all know, that's not equally available on a global scale. MALDI-TOF is the gold standard for bacterial ID in Western countries now, but there is still a need for cheaper, smaller, more rapid, even more simple equipment that covers a broader range of organisms, and that might also include the analysis of smaller molecules, and then this work done by Caroline and Adrian Agley in Switzerland, where data is being integrated from diverse perspectives. I think that is a development that we should very carefully monitor over years to come. Still, mass spec is not the only diagnostic gold standard technology. PCR and sequencing and other omics technologies will enter the routine lab, but as a final word, I think that the more data we would be able to put into our predictive diagnostic modules, the better and the more clinically valuable these outcomes are going to be. And combining diagnostic data with clinical data in the big data format will become more commonplace in the years to come. And this is where I want to stop, while thanking my colleagues for providing slides, discussions, and a pleasant working environment in the southern part of France. Thank you.
So thank you very much, Alex, for this really nice presentation. I think it's just the perfect presentation to close this really first session of the symposium. So of course, there's questions for you, as you can imagine. So just let me go. So the first one is from Valerie, and she's asking, has the use of Malditel help with identification of an association of certain organisms with clinical conditions, like, for example, acne, hidradenitis suppurativa? Sorry, I don't know this name. So, and pilonidal sinuses.
Sorry, I didn't hear the question very well.
Oh,
is it
so
in
the
chat?
You can read it normally. It's has the use of Malditel helped with identification of association of certain organisms with clinical conditions?
I see the question now. It's usually the other way around. It's not like that MALDI identifies a pathogen, because most of the pathogens are well known, and for pathogen identification, you need culture, and you need some of more basic technologies, and the group of DJ Raoult, for instance, have shown that certain Bacteroides species are more prevalent in colitis in neonatal intensive care, in pediatric neonatal intensive care settings. And there they identified the bug by doing a culturomics approach, but then for the characterization and for the species identification after that first discovery, mass spec is obviously the perfect tool. But I have no idea whether sort of a comparative mass spec approach has been used to really identify new pathogens.
Okay, we also have another question from Belen, who just spoke a bit earlier today. Can the rate of Bacteroides firmicutes be calculated using Malditel?
That's a good question. My feeling is that everything that you do with mass spec and that has a real hard quantitative outcome has to be taken with serious doubts and considerations, because you know it strongly depends on the sample processing, on the let's say the extractability of strains, and the ionization efficiency in the process. So This is where I'm a key opinion speaker and not that much an expert at this type of thing. I'm sure that some of my colleagues could provide you with more precise answers, but my take would be that this is something that you would have to take care of, that you would have to sort of pre-assess before you start doing this routinely. And I don't know whether we're capable of this right now.
Okay, also we have some comments from Ray and also from Valerie, so you can read it a bit later because it's really long comments. We also have a last question. Is Biomarkers investigating tools for the integration of mass spectrometry data in an open access fashion? Thanks, Michael.
Michiel Bekskens, I know the name. He used to be in the same lab where I worked like 10 years ago. Ik hoop dat het je goed gaat, Michiel. In open access fashion, yeah, we're a company, yeah. So open access is always a very complicated issue, and you know we do have software which allow the customers to build their own databases, and then it's up to you whatever you do with it. And so there is an option to do that using the Biomarkers equipment. The other thing is that if you use the mass spectrometry for microbial identification, you have to go through FDA, the federal drug, the US FDA for validation and for acceptance. The moment you do that, the database cannot be touched anymore. The moment you start modifying your database, you'll have to go through FDA approval again in order to make sure that you're not screwing up. And that also strongly limits our options for sharing databases. But then again, if you have a specific question and you have a bug with a very bizarre spectrum and you want to check that out, then you can always contact us and we'll try to help you. Which in your case will often be the case, I guess.
Okay, thank you very much, Alex. I think we do not have any more questions for you. So once again, thank you very much. It was a really, really interesting talk. I'm sorry that you were not in the really first lane of the early symposium, of course. But I think it was a good way to close the first session. You just put in the real reality of mass spectrometry and diagnostic right now.
Okay, thank you.
Thank you to you. So apparently we just reached the end of the first session of our symposium. So I would like to thank all the speakers. So I don't know if all the speakers are still here, can switch on the camera just to say a last goodbye. For tomorrow, we will also have more talks about parasitology in general with other institutions, and also more talks about a company like Biomarkers with Kathleen Vangs and from Brugge with Marcus Stimk. So keep aware of tomorrow's session. It was my pleasure to host these first sessions. So thanks again to all the speakers, and a big thanks to all the people who just attending to the symposium and people who ask questions. Thank you very much. And for tomorrow, see you tomorrow. Bye.